Okay, so we're going to start our blood notes. Just pulling up the right slide. There we go. All right, so blood and blood spatter. Blood spatter is the last thing we do in this unit, so it is going to be a little while till we get to the blood spatter and the blood spatter labs. But we will get to it in this unit. First is going to be more about a little bit about history, blood composition, learning how to use blood typing, so like determine if it's an A, B, O type blood, how it's used in crime scenes, how blood determination is possible. So like if I know that like the child of my suspect is O negative, then I would know there's like only a certain number of blood types my suspect could be if I know the child's blood type. So how to like kind of work your way back from inheritance. So we'll do kind of all that stuff first, how to test to see if something is blood at a crime scene or if it's just like red paint, something like that. Sorry. And then the last thing that we'll do is blood spatter. So looking at blood spatter on the wall and see kind of what it could tell you a little bit. All right, so here we go. So the history of blood, we're not going to talk very much about it. I just do like to mention kind of how far we come and like the leaps and bounds we've made recently. We spent a lot of time doing weird stuff with blood for just thousands of years, not understanding it at all. And then finally, we kind of start figuring some stuff out and kind of like all in a row are getting more technology surrounding our ability to analyze blood. So the practice of bloodletting began around 2500 BC. That's before the time frame that we're in right now. So it's 2021. So that's 2021 years. And then that's 2500, so even pre that. Okay. So like we're talking really, really long time ago. But they're still doing bloodletting. And they did bloodletting till the 19th century. So that was, I mean, not recently, but I mean, still like from forever ago until the 19th century. So they continued thinking that just releasing blood was a good idea for a really long time. I'm just surprised that it lasted that long. In 500 BC, so again, that's pre the 2021 years that we're in now, it's before that. Greeks distinguished between arteries and veins. So they already knew that we had different types of vessels in our arms, one heading towards the heart and one heading away. And even though they learned that, they still did so many weird things, continuing on for thousands of years after that, even though we already knew we had those arteries and veins. They still just understood so very little about blood. So remember, arteries are the ones that are carrying oxygenated blood from the heart to your body. So they're the ones leaving the heart that have been reoxygenated after leaving the heart and the lungs and going towards your body. Everything that's heading back up or back in or back down, like if they're heading back to the heart to become reoxygenated by the lungs. So those are veins. Anton van Leeuwenhoek, hard to say that. I'm sorry, guys, it's late. Viewed blood cells under a microscope for the first time in 1659 AD. So now we're in our current time frame here. So 1659, we're into 2021. So I mean, still 400 years ago. But I mean, like, I'm pretty impressed that they had a microscope and could already see blood. So seeing these are what our red blood cells look like, just little red circles where human blood cells are pretty round. And then these little globs here are our white blood cells. You can already see that there's a lot less white blood cells than there are red. First blood transfusion was done in 1795. And you kind of seen a little clip of that yesterday with our warm-up about kind of the struggle with transfusions, how they tried it with animals first and had a couple successes, oddly enough. And then people started dying and they couldn't figure out why until we figured out blood types after this. So 1795, we still didn't know about blood types. So people would continually die after blood transfusions, and you wouldn't really be sure why until 1901. Carl Landsteiner discovered that there were different blood types, that the reason why we reacted badly was that if you have an A blood type and I give you a B blood type, oh my goodness, you could definitely have an adverse reaction. Not everyone that gets the wrong blood type in a blood transfusion would die. Sometimes you would just get really sick, but you definitely can die. Alfred de Castellello discovered the fourth blood type, the AB. So Carl Landsteiner is the one that we're going to kind of give credit for finding the blood types, but he didn't find the AB. It took another year for Alfred de Castellello to figure out that there was another one that actually did react just slightly differently. So let's kind of already talk about our blood types here just a little bit. So if you have an A blood type, that means you have, so like notice that they're all red blood cells. Like we all have red blood cells. All humans, all mammals have red blood cells, okay? So we have these red blood cells, but they're these little proteins. They're called antigens that are on top of your blood cell that tells us your blood type. So it's this additional protein that is just genetically coded to sit on top of your red blood cell. And if you have the particular shape that is A, then you're type A. If you have a different shaped antigen that is sitting on your blood cell that is a shape of B, then you're type B blood. If you have both of those, you have both those proteins called antigens sitting on top of your blood cell, then you're AB because you have A and B. And if you're O, I like to think of as zero, meaning it has zero antigens on its cell. So it doesn't have A or B. It has no proteins on its cell. So that's why it's type O, is because it doesn't have antigens on its cells. Now, where the problem comes in is our body has an immune response. Our body fights what we don't recognize. Just like we fight off viruses, your blood cells would fight off blood cells that your body thinks are foreign. So how determines it foreign if it doesn't have the same protein on it? Okay. So if you are type A, then the things you have in your plasma, your immune response would fight things that are shaped like B. So if I gave an A blood type person B blood, their blood would respond and attack it. Now, that attacking would cause clumping. Now, the antibodies would try to break down that blood, saying it's not supposed to be here. They try to break it down and get rid of it, and it might accomplish this task, get rid of it, and you get better. But it might also clump up so much that it causes a blood clot, which would go to your heart and your brain. So heart attack, brain, stroke, and can also be broken down, creating toxic substances that your kidney can't process, so causing kidney failure. So a lot of bad things can happen with giving the wrong person blood. And it's based on, if you have type B, then you have A antibodies. You have things that would fight off A blood. If you have AB blood, well, your body's not going to fight A or B because then it would fight itself. So it has no antibodies, which means if you have type AB blood, I can give you any kind of blood and your body won't fight it because your body doesn't have antibodies floating around in it. It accepts all types. So if you're an AB blood type, you are the best person to be a receiver. Okay, so you are the best receiver. You can receive pretty much all kinds of blood. And then group O, oh no, what just happened? If you're type O blood, well, you don't have A or B on your blood cell. So your body doesn't recognize A or B. So your body fights A and B blood. So you can't have either. You can't have A blood, you can't have B blood, you can't have AB blood. The only blood you can have is O. So the group O is the hardest to give blood to because they can only receive other O blood types, but they are the easiest to donate because being that they have nothing on them, these blood types won't fight it because there's nothing on it to fight. Now, if that was a lot of information, we're going to explain it more and more detail as the days continue. I just wanted to go ahead and preface it with these pictures. All right, then we're moving up. So that was early 1900s. We finally figured out blood types, start doing a little better with blood transfusions. In 1922, Percy Oliver established the first blood donor service. So people being actually able to go in and donate blood on a regular basis. In 1935, the Mayo Clinic developed a method to store blood for transfusions. And then Dr. Bernard established the first blood bank in 1937. So it wasn't until the 30s that we had blood on hand. So if you think about that, I think a lot of us feel really comfortable in the fact that we have blood, that if you were bleeding out, you could go to the hospital and you could get blood. It's really pretty recent that that's been a possibility. Because this is when it started. So do you think every city everywhere had blood? No, absolutely not. It took a long time, probably another 50 years for you to be pretty sure you could get blood. And then they still might be out of your type. Blood shortages do happen, actually still happen all the time. So I'm going to use this reminder, please give blood if you can. We have shortages of O and O negative, O positive and O negative all the time because it's the most used blood. And any of the negative blood types are hard to come by. So if you have a negative blood type, you're a really good donor. But if you're any kind of blood type, it would be helpful to donate your blood. All right, 1940, Carl Landsteiner again. So he's the one that found the blood types. He then discovered the Rh protein. So the Rh protein is called the rhesus factor, which is why it's called Rh, because rhesus. It came from this rhesus monkey. That's where they found it. So it was first discovered in the rhesus monkeys in India, often used for experimentation. Sad. The importance of the Rh factor. So something they had noticed was that they were having this issue. It happened pretty often back then. Okay, so we're 1940, everywhere down. Women would have a hard time giving birth to a live baby. Like they'd have a lot of miscarriages. Okay, so I mean, not that miscarriages don't still happen, but much more common. And something that could have caused it, something they figured out in this time frame, is actually about blood types. So an Rh negative woman. So what that means. So the Rh protein is just another protein that sits in your cell. So if I go back for a second, it's another protein that sits in your cell. They didn't find it yet. They found the A, they found the B, but they didn't find the Rh. The Rh is another protein that if you have it sitting on your blood cells, you're positive. So like if you hear A positive, B positive, or O negative, B negative, if you have it, you're positive. You have an A positive blood type if you have the A antigen on your cells and you have the Rh. So the Rh makes you positive. If you don't have the Rh antigen, then you are negative. Okay, so if you just don't have it, you're negative for not having it. So they were finding it. They were finding out there were these women that were negative blood type, which means they don't have the Rh protein, would become pregnant with an Rh positive. So their fetus had an Rh positive blood type, which can happen if the father had it, and that's the genes that got carried on. They could have a positive blood type. That sometimes her body that was negative would not recognize that positive protein, which remember, it's not positive, it's not like a charge, it's just the Rh protein. So the Rh protein that's on them, it wouldn't recognize that and basically would fight the baby and kill it. Okay, so like her body would continually cause her to have miscarriages because it didn't recognize that blood type. Now, we've actually figured out some medicine women can take. So if you know that you are a negative blood type, this will just be something that your doctor will have to bring up later. If you're pregnant, that they will have to check it and you will have to take medicine. Especially if it's your second. If it's your second baby, it will matter even more because you usually fight it after the body recognizes it like the second time around. The first time around, it may not recognize the difference. So anyways, that's the first time we found the positive and negative gene. The positive negative on blood cells, which just means they have the Rh protein or not. In 1959 was our first case of AIDS, and that just comes up because it was actually a really big deal. Like for us, like I feel like we're just like, okay, sexually transmitted disease we've known about since we were born, but they had it. So like if I go back to like my dad's age, them finding out about this disease, it was a blood disease you couldn't get rid of, was actually this gigantic deal. In 1984, they identified the virus causing AIDS. It was like that long. In between time did they actually notice that we had it here. Okay, so it was in the 80s when AIDS actually became something to worry about. It. So kind of pre the 80s, people were a lot less careful with sexually transmitted diseases because they hadn't had anything that was that life-threatening before. And now I feel like we're so much more knowledgeable about that. But still, like, I mean, it's been recent that we really realize there's so much more bloodborne pathogens than we thought before. In 1985, the development of the ELISA test is to screen for diseases such as HIV. So now we have tests that can screen for blood diseases. In 1987, scientists began developing blood screen tests for other infectious diseases other than AIDS. And then in 1989, Sir Alec Jeffrey discovered DNA. So we've talked about that one before. Huge thing. First case solved of DNA and forensics was in 1990. That's 31 years ago. Okay, not even really just 30 because we just started 2021. So I mean, this whole process of us being able to find blood and take DNA from it is very new. Like, yes, that was the first case, but you think like it was widespread and everyone used it? No, it takes at least 15 to 20 years for things to become widespread, and this was only 30 years ago. So honestly, DNA is still kind of in the works. All right, now we're going to talk about serology, just a couple of vocab terms. Serology is the study of body fluids. So that's the unit we're in. we're focusing on blood, but any kind of body fluid would fall into this category, a person who studies body fluid. So you could study blood spatter. That's its own little sector. Okay, blood spatter patterns can be used to reconstruct a crime scene and show how an attack was made. Blood can connect an object with a crime. Obviously, if you find blood on the object and you found the same blood at the scene, blood can connect a suspect with a crime. So if there was blood on the suspect, if the suspect's blood was left at the scene, okay, so all those things can place a suspect at the crime scene. Blood typing. So what we're going to learn how to do first is how to tell what blood type you are. So O positive, A positive, B positive, that kind of thing. How to tell what type you are is always class evidence. Because remember, if I get to a crime scene, I find some blood, and I do the blood typing test, so we'll learn how to do. It's actually pretty quick and pretty easy. And I figure out that my suspect is, or the blood at the crime scene is B negative. And my victim is A positive. Okay, so I know it's not the victim, but there's other blood here that is B negative. Now, if the first suspect I find, like they don't have an alibi, the victim picks them out in a lineup, like that whole thing, and they are B negative, does that automatically mean that I have them, that they're it? No, it is class evidence. Class evidence means I'm not for sure it's that one person. I'm just sure that your suspect does fall into this group of people that is B negative blood type, which is only 1.5% of the population. So 1.5% of the population. So that'd be narrowing us down quite a bit. Now, and if the suspect was O positive, that's a much bigger pool of people. That's 39% of the US population. So almost 40% of our population is O positive. So that'd be narrowing us down not as much, like almost half. So obviously these would be more helpful. But anytime you are doing blood typing, it's not going to single you out to giving you one person. It's just going to rule out suspects and add to the case. Now, if you can do DNA profiling, remember it's more expensive, takes more time. Most cases will only do it if they're pretty certain they have their guy and they have the money. DNA profiling from blood is considered individual, so it will tell me for sure, like, this is the person, okay? Because we've matched their DNA to the blood of their scene, so it is individual evidence that can help us pinpoint the suspect, not a group of suspects. All right, so just a little bit about the composition of blood. I don't know how much y'all know about your blood. If you've given blood, you can actually watch them separate it into these three categories. It's pretty interesting as long as you're not squeamish. But as they are taking your blood, it will be taken off to the side and it will be separating, like right there. You can see them separating usually in almost every place I've went to. I can watch them separating the blood bags right there. And they separate it out into your plasma, your platelets, and your red blood cells. The red blood cells tend to look not red. They look reddish brown, like really dark, way darker than you would expect. And then the platelets look clear. And then your plasma does look like this yellow pea color. It's very unsettling. So 55% of your blood is plasma. The white blood cells and the platelets are tiny little percent. So less than 1% of your blood is that. So way less of that white liquid you would see. And about 45% is just red blood cells. If you didn't know, when you donate your blood, they actually only give people the red blood cells. They don't give them the plasma or your white blood cells, because that can sometimes just have more adverse effects than actually being helpful. And the red blood cells is actually what helps you generate more blood. So is the best thing to donate. So when you donate blood, they will take your blood out, they will separate it into these three parts, and then they will only keep the red blood cells, and the rest of it is just thrown out. Sometimes they use the plasma for other things, but basically they just use the red blood cells. And no, they don't just give it back to you. Sorry. Blood is a tissue that circulates around through the body. The average adult has about 10 pints of blood, which I'm pretty sure, if I remember correctly, it's about a gallon and a half. So like that's kind of something to think about, how much blood you actually have in your body. It's about a gallon and a half. The bigger you are, the more blood you have. If you're really tiny, then you're probably more like a gallon. If you are like a giant football dude, maybe getting closer to 2 gallons. But that's kind of the sway. It's like a gallon to 2 gallons of liquid in our bodies, depending on how big you are. Blood is made of both cells, which is the red blood cells and the white blood cells and the platelets. Those are all cells. and then plasma, which is mostly water. I'll talk about that in a second, which is the other 55%. So blood cells are made in your bone marrow. So up here in your bone marrow is where your blood cells are created. The spongy material in the center of the bones, the body will replenish the elements given during blood donation, some in a matter of hours and others in a matter of weeks. So it kind of depends on you how fast your body will replenish your blood cells, how fast it makes it. But some people pretty quickly, you'll already be making more blood cells, and some people will take longer, could take up to several weeks for you to get your blood cells completely back. Now we're going to talk about the 45%, just the cells first. So out of the 45% of your blood that are cells, there's white, red, and platelets. The red blood cells are called erythrocytes. They have hemoglobin. They carry oxygen. They are iron containing, and they have no nucleus. So when we see them under a microscope later, which we will, they're just a red circle. There's no nucleus. There's nothing inside them to see. The reason why they're pretty sure that humans and other mammals have evolved to do this, because all mammals basically have red blood cells like this that don't have a nucleus, which is pretty weird. Most cells have a nucleus, but red blood cells don't, is to make more room for more oxygen. So it just makes us to where we can carry more oxygen around with us, makes our bodies more efficient. And the iron and the hemoglobin is what is making it that red color. Now, the white blood cells are called leukocytes. They're the immune system cell. They fight disease and produce antibodies. So the antibodies that fight off viruses or fight off the wrong kind of blood, if you get it, they contain DNA. Notice red blood cells don't contain DNA because DNA is contained in a nucleus. If they don't have a nucleus, they don't contain DNA. So white blood cells do have a nucleus. See these white blood cells? They have this, funny shapes in the middle. That's their nucleus, and that's where the DNA would be housed. The two most common types are lymphocytes and neutrophils. There's quite a few types of white blood cells. They do different things. They fight off different things, but they all have your DNA inside them. And then lastly, there's platelets, are called thrombocytes. They are small cell fragments. So they're not even a full cell. They're cell fragments, and they're used to help blood clotting. So if you have like a cut, then they'd go and start doing their clotting thing, which is why you don't need that much of them in your blood. If you're ever sick, you have more blood cells, more white blood cells in your bloodstream because you're fighting off a virus. So your body creates more white blood cells to fight off the virus. But normally, we have less than 1% of white blood cells and platelets, and the rest of it is all red blood cells. So when you think about that little tiny bit, there are about 1 billion red blood cells in 2 to 3 drops of blood. For every 600 red blood cells, there are about 40 platelets and 1 white blood cell. 600 red blood cells, 1 white blood cell. And the 1 white blood cell is the only thing that contains DNA. So when I mentioned in the past that DNA is harder than you would think to do, you're just like, oh, I'm bleeding, do a DNA test. Most of your blood has no DNA in it. Only 1% of your blood has DNA in it. So if I only get a tiny drop of blood, I may not have enough white blood cells in that drop to actually do a good DNA test. So that's why it's like you really do kind of have to have a sufficient amount. It has to be fairly new, well preserved. All those things have to be true for us to be able to do a DNA test. Okay, almost done. Yes, 2 slides. So the blood plasma is the yellowish liquid. Okay, it's not yellowish liquid above. So no cells. It's just 90% water, mostly water. The other 10% are dissolved proteins, antibodies, hormones, clotting factors, so just kind of other stuff floating around in there. Nutrients like oxygen, sugars, amino acids, salts, minerals, and waste. So as we are also getting rid of our waste as the blood continues to. So all that stuff floating around your plasma. So basically just with all that liquid, it just helps it move. Helps it move through your body because that's what it has to do. It has to take oxygen to your cells, has to take sugar to your cells, has to take amino acids to your cells. So all those things your body needs, it takes it there. And then lastly, how does it take it there? Through our vessels, obviously. So we have arteries, and I've already said it, carries blood away from the heart. They divide again and again and eventually form very tiny vessels called capillaries. So as we're getting to our teeny parts, like your fingers up here, we're going to have capillaries, giving them the nutrients they need as well. And as they have given them nutrients, as they come back, they will take the waste. So anything from leftover reactions that we no longer need, you're getting rid of the waste, going back to the liver to be clean, going back up to the heart to be reoxygenated. So the capillaries gradually join up with another to form large vessels called veins. Veins carry blood back towards the heart and the lungs to get reoxygenated. At any given moment, about 30% of your blood in your systematic circulation we found in your arteries. So about 30% is actually being actively pumped to your body to be helpful. About 5% is in your capillaries, so in the small pieces. And about 65% is just heading back to the heart. About 65% is just going back to your veins. We'll need to know more about veins and arteries later, about like how it would look if you cut an artery compared to if you cut a vein. That's all you need to know for now. So there we go. We'll stop there.